For decades, scientists assumed that once modern humans left Africa, the story of human evolution basically stopped, and everyone outside Africa carried roughly the same genetic package. Then researchers actually started comparing full genome sequences between Chinese and European populations in detail, and the results overturned that assumption completely. Buried inside the DNA of these two populations were traces of a different human species entirely, evidence of separate evolutionary paths and genetic adaptations so specific that scientists could pinpoint almost the exact region and time period where certain traits first appeared. What they found didn't just describe two populations. It rewrote the timeline of everything that happened to our species after we left the African continent. To understand why this comparison matters so much, you have to go back roughly 70,000 years to the point when small groups of modern humans first began migrating out of Africa and spreading across the rest of the planet. For a long time, scientists pictured this as a single, relatively simple journey outward. Genetic evidence tells a far messier story. The ancestors of modern Europeans and modern East Asians appear to have split from a shared population somewhere between 40 and 50,000 years ago, likely somewhere in the middle, East or Central Asia, before heading in dramatically different directions. One branch pushed westward into the harsh, cold landscapes of Ice Age Europe. The other pushed eastward across Asia, eventually reaching what is now China, and later crossing into the Americas entirely. That single split, tens of thousands of years ago, set the stage for every genetic difference researchers would eventually uncover. The first major shock came from an entirely different species altogether, when researchers finally sequenced a complete Neanderthal genome and compared it against modern human populations. They discovered that virtually everyone with ancestry outside Africa carries a small percentage of Neanderthal DNA, roughly 2% on average. That alone was a stunning finding, confirming that modern humans and Neanderthals interbred, rather than simply competing and replacing one another as separate species. But then researchers noticed something even stranger buried in the comparison between Chinese and European genomes specifically. East Asian populations consistently carry meaningfully more Neanderthal DNA than European populations do, with some estimates placing the difference as high as 20% more Neanderthal ancestry in East Asians compared to Europeans. This finding genuinely puzzled scientists for years because it doesn't match the geography of where Neanderthals actually lived. Neanderthals occupied Europe, the Middle East, and parts of Central Asia. But they never set foot in East Asia as far as any fossil evidence shows. So how could modern populations in China end up with more Neanderthal DNA than the modern population living in the exact region where Neanderthals actually existed? Researchers eventually proposed that the ancestors of East Asians experienced a second, separate wave of interbreeding with Neanderthals after splitting off from the ancestors of Europeans, essentially picking up an additional dose of Neanderthal genetics on their journey eastward that European ancestors never received. A more recent study, published in 2023, offered an even more compelling explanation for this Neanderthal discrepancy, one that had nothing to do with a second interbreeding event at all. Researchers analyzing ancient DNA spanning 40,000 years found that early European hunter-gatherers actually carried Neanderthal ancestry levels similar to those found in East Asians. The difference only emerged later, when a large wave of Neolithic farmers migrated into Europe from Anatolia roughly 10,000 years ago, carrying comparatively less Neanderthal ancestry themselves and effectively diluting the Neanderthal genetic signal already present in the European population. East Asia never experienced an equivalent large-scale replacement migration, so its Neanderthal ancestry simply remained more concentrated over time. In other words, the difference wasn't really about who interbred with Neanderthals more. It was about which population later got genetically diluted by an entirely unrelated migration thousands of years afterward. Neanderthals aren't the only ancient relatives whose fingerprints show up unevenly between these two populations. Denisovans, a mysterious archaic human species identified almost entirely through DNA extracted from a single finger bone found in a Siberian cave, left a genetic legacy that shows up far more strongly in populations across Asia and the Pacific than it does in Europe. While the direct Chinese population carries relatively modest Denisovan ancestry compared to groups like Melanesians, researchers studying East Asian genomes have still found specific Denisovan-derived genetic variants tied to real biological outcomes, including certain immune system responses, and disease risk factors that simply don't appear in European genomes at all. One recent study even found that Denisovan-derived genetic variants in East Asian populations were specifically linked to differences in coronary artery disease risk, a connection entirely absent when researchers ran the same analysis on European genomes. If you're finding these genetic detective stories as fascinating as we do, hit that subscribe button, because uncovering exactly how ancient interbreeding shaped modern Populations is exactly the kind of mystery we love digging into. Beyond ancient interbreeding with other human species, researchers have also identified specific genetic mutations that arose independently within East Asian populations after the split from European ancestors, mutations tied to visible physical traits that most people simply assume are unrelated genetic coincidences. One of the most well-studied examples involves a gene called EDAR, responsible for regulating the development of hair follicles, sweat glands, and several other skin features. Scientists traced a specific variant of this gene, known as EDAR370A, to a single mutation that most likely arose in central China roughly 30,000 years ago. This one genetic change is now believed to be responsible for several traits common throughout East Asian populations, including thicker hair strands, a distinctive tooth shape, and a notably higher density of sweat glands, verified in laboratory studies comparing Han Chinese volunteers against populations carrying the ancestral version of the gene. What makes the EDAR discovery so remarkable is how directly researchers were able to confirm its effects, moving beyond simple statistical correlation into genuine experimental proof. Scientists engineered the exact same mutation into laboratory mice and observed strikingly similar changes, including denser fur and increased sweat gland density in the modified animals. This gave researchers rare, direct experimental confirmation that a single identifiable mutation arising in one specific ancient population at one specific point in history was responsible for physical traits still visible in 30,000 years of descendants today. Very few genetic differences between human populations come with this level of experimental certainty attached to them. Skin pigmentation offers another genuinely surprising twist in this comparison, one that challenges a common assumption most people carry without ever questioning it. Many people assume that lighter skin evolved just once in human history and then simply spread outward from a single origin point. The genetic evidence tells a completely different story. Researchers studying pigmentation, genes have found that European and East Asian populations evolved lighter skin through largely separate genetic pathways, a phenomenon scientists call convergent evolution. Europeans developed lighter pigmentation, primarily through variants in genes called SLC24A5 and SLC45A2, while East Asian populations achieved a broadly similar outcome, lighter skin better suited to lower ultraviolet light environments, through an almost entirely different set of genetic variants, particularly within a gene called OCA2. This means that if you compared the specific genetic code responsible for light skin in a European person against the specific genetic code responsible for light skin in a Chinese person, you would find largely different mutations producing a visually similar result. Evolution essentially solved the exact same environmental problem, reduced sunlight requiring reduced pigmentation for proper vitamin D synthesis, twice independently, using two largely different genetic toolkits scattered across two different populations that had already been separated for tens of thousands of years. Researchers studying this pattern describe it as one of the clearest documented examples of convergent evolution found anywhere in the modern human genome. Diet-related genetic adaptations reveal yet another area where these two populations diverged sharply after their ancestors went their separate ways. Consider lactase persistence, the genetic ability to continue digesting lactose, the sugar found in milk, well into adulthood. In most mammals, and in most humans historically, the ability to digest lactose shuts off naturally after infancy. Populations with long histories of dairy, farming, concentrated heavily in parts of Europe, evolved genetic mutations allowing lactase production to continue throughout adulthood, a trait now found in a majority of people with European ancestry. East Asian populations, lacking the same multi-thousand-year history of large-scale dairy farming and cattle domestication, never developed this mutation at nearly the same frequency, meaning lactose intolerance in adulthood remains dramatically more common throughout East Asian populations today compared to European populations. Alcohol metabolism tells an equally striking story running in the opposite direction. A specific genetic variant affecting an enzyme called ALDH2, responsible for breaking down alcohol byproducts in the body, appears at dramatically elevated frequencies specifically within East Asian populations, found in an estimated 30 to 50% of people with Chinese, Japanese, or Korean ancestry, compared to a small fraction of 1% among people of European descent. This particular variant causes an inefficient buildup of a toxic compound called acetaldehyde during alcohol metabolism, producing the well-known facial flushing reaction, along with nausea and rapid heartbeat, that a significant portion of East Asian individuals experience after drinking even modest amounts of alcohol. Researchers believe this variant likely became common in East Asia due to complex historical factors involving rice cultivation, fermentation practices, and possibly even protection against certain parasitic infections common in ancient rice farming communities. Beyond hair, sweat glands, and skin tone, researchers have identified an entire cluster of additional traits linked to genetic differences between these two populations, some of which sound almost trivial on the surface, but turn out to trace back to fascinating evolutionary stories. Consider earwax. Most people never think twice about whether their earwax is wet and sticky or dry and flaky, but researchers discovered this difference traces back to a single gene called ABCC11. The variant responsible for dry earwax appears at dramatically higher frequencies in East Asian populations compared to European populations. Scientists believe this same gene variant is also connected to reduced body odor production, and some researchers suspect the mutation may have spread alongside other adaptive traits during the same general period when populations were adapting to the colder climates of ancient Northeast Asia, though the exact evolutionary advantage remains an active area of research. Eye shape offers another visible trait, with a genetic story still being pieced together by researchers today. The epicanthic fold, a fold of skin covering the inner corner of the eye common throughout East Asian populations, has long been assumed to have a straightforward genetic basis. Yet scientists have struggled to pin down the exact combination of genes responsible with the same precision achieved for traits like the EDAR-linked hair and sweat gland changes. Some researchers have proposed that the epicanthic fold may offer protective benefits against cold, dry wind exposure, potentially explaining why the trait became so common throughout populations descended from ancestors who spent extended periods in the harsh climates of ancient Siberia and Central Asia before eventually settling further south and east. Altitude adaptation provides one of the most dramatic examples of population-specific genetic change found anywhere in modern human genetics, and it further illustrates just how differently evolution shaped populations after their ancestors parted ways. Populations living on the Tibetan Plateau, closely related to broader East Asian ancestry, carry a genetic variant in a gene called EPAS1 that allows their bodies to function normally at extreme high altitude without producing the dangerously thickened blood that low-altitude populations develop when exposed to the same oxygen-thin conditions. Remarkably, researchers traced this specific variant directly back to Denisovan ancestry, meaning modern Tibetan populations inherited a genuinely life-saving adaptation directly from interbreeding with an extinct, archaic human species tens of thousands of years ago. No equivalent adaptation exists within European populations, since European ancestors never faced the same sustained evolutionary pressure of surviving permanently at extreme altitude. Taste perception adds yet another layer to this genetic comparison, particularly around the ability to detect certain bitter compounds. Found in vegetables like broccoli, cabbage, and Brussels sprouts, researchers studying a specific bitter taste receptor gene called TAS2R38 found that the frequency of taste-sensitive versus taste-insensitive variants differs measurably between Chinese and European populations, helping explain why perceptions of bitterness in identical foods can vary so significantly between individuals from different ancestral backgrounds. While this might sound like a minor curiosity, perception genetics actually plays a meaningful role in dietary research, since populations that perceive certain vegetables as more intensely bitter often show measurably different consumption patterns of those foods, a factor increasingly considered in nutritional and public health research. The immune system reveals some of the most medically significant differences researchers have documented between these two populations, extending well beyond the Denisovan-linked coronary disease connection mentioned earlier. Certain variants within the HLA gene complex, a group of genes responsible for helping the immune system distinguish between the body's own cells and foreign invaders, show substantially different frequency patterns between Chinese and European populations, patterns that researchers believe reflect thousands of years of exposure to different regional disease environments. These HLA differences carry real-world medical significance today, particularly in the field of organ transplantation, where matching donor and recipient HLA types across different ancestral populations remains measurably more difficult than matching within the same population, a persistent and well-documented challenge for patients from ethnic minority backgrounds, seeking transplants in countries where the donor pool skews heavily toward a different ancestral majority. Researchers have also traced measurable differences in genes related to how efficiently different populations process certain classes of pharmaceutical drugs, an area of study known as pharmacogenomics that has become increasingly important as personalized medicine advances. Variants within genes responsible for metabolizing certain blood-thinning medications, chemotherapy drugs, and even common pain medications appear at meaningfully different frequencies between Chinese and European populations, sometimes requiring genuinely different dosing guidelines to achieve the same therapeutic effect safely. This is far from a purely academic distinction. Incorrect dosing, based on genetic assumptions drawn primarily from European population studies, has, in documented cases, led to serious adverse drug reactions in patients from different ancestral backgrounds, driving major pharmaceutical companies and regulatory agencies to increasingly require more genetically diverse clinical trial populations before approving new medications for widespread use. None of these genetic differences imply anything whatsoever about intelligence, character, or human worth. And researchers studying population genetics are consistently clear on this point precisely because this kind of research has been badly misused throughout history to justify discrimination and pseudoscientific racial hierarchies. What these differences actually represent is something far more interesting scientifically, a detailed genetic record of how two populations, descending from the exact same African ancestors, adapted independently to wildly different environments, climates, food sources, and disease pressures across tens of thousands of years of separate history. The traits researchers have identified relate specifically to things like skin pigmentation suited to different sunlight levels, digestive adaptations suited to different historical diets, and immune responses shaped by different disease environments, not to anything resembling cognitive or behavioral differences between populations. What makes comparing Chinese and European DNA scientifically valuable goes well beyond satisfying historical curiosity about migration routes and ancient interbreeding events. Researchers studying genetic differences between these two populations have made real, practical medical discoveries along the way, particularly around how specific genetic variants influence disease risk and drug response differently across populations. Certain medications metabolize differently depending on which population-specific gene variants a patient carries, and pharmaceutical researchers increasingly recognize that drug dosing guidelines, developed primarily through studies on European populations, don't always translate accurately to patients with different ancestral backgrounds, including East Asian populations specifically. This has driven a broader push within medical genetics toward more geographically and ancestrally diverse research, ensuring that genetic discoveries benefit all populations rather than just the ones historically overrepresented in scientific studies. The migration timeline itself has also grown considerably more detailed thanks to advances in ancient DNA sequencing over the past decade, allowing researchers to move well beyond broad estimates and into genuinely precise reconstructions of population movement. Ancient DNA extracted from human remains found in Siberia, dated to roughly 45,000 years ago, has helped researchers pinpoint the approximate window during which the ancestors of modern Europeans and modern East Asians were still genetically indistinguishable from one another, sharing the same recent common ancestors before beginning to diverge along separate paths. This same ancient genetic evidence has allowed scientists to trace the specific route East Asian ancestors likely followed, moving through Central Asia before splitting further into populations that eventually settled across what is now China, Japan, Korea, and Siberia, with some groups continuing onward across the Bering Land Bridge into the Americas. This level of genetic detail has also allowed researchers to resolve long-standing archaeological debates that historians and anthropologists had argued over for decades, using nothing more than physical artifacts and skeletal remains. For years, scholars debated whether early populations across East Asia represented a single, continuous wave of migration or several genetically distinct waves arriving at different points in history. Genetic. Sequencing has now confirmed that East Asia experienced multiple distinct migration waves over tens of thousands of years, with later populations sometimes genetically replacing or heavily mixing with earlier ones, a level of demographic complexity that would have been essentially impossible to reconstruct through archaeology alone. Comparative genetic research between Chinese and European populations has also revealed something researchers describe as remarkably low genetic diversity when measured against certain African populations, an important and often overlooked finding for anyone trying to understand the bigger picture of human genetic variation. Because both European and East Asian populations descend from a relatively small group of migrants who left Africa within the last 100,000 years, both groups actually carry noticeably less overall genetic diversity than populations that remained within Africa throughout that entire period. This means that, in a very real genetic sense, the differences separating Chinese and European populations from each other are considerably smaller than the genetic diversity that exists within many African populations alone, a finding that consistently surprises people encountering population genetics for the first time, and one that further undermines any notion of rigid, deeply divided human racial categories. Modern large-scale genome sequencing projects, including efforts specifically focused on generating comprehensive reference genomes for East Asian populations, continue refining this picture even further, correcting earlier genetic databases that were historically built using research samples drawn overwhelmingly from people of European ancestry. This historical imbalance in genetic research meant that for years, scientific understanding of human genetic variation was skewed by an incomplete and disproportionately European dataset, a gap researchers are now actively working to close through expanded international sequencing initiatives covering East Asian, African, and other historically underrepresented populations. As these more comprehensive datasets continue to grow, researchers expect the comparison between Chinese and European DNA to keep revealing new adaptive traits, new disease risk factors, and new pieces of the ancient migration puzzle that remain hidden within the genome for now, simply waiting for sufficiently detailed sequencing technology to bring them into focus. The story these genetic comparisons tell, ultimately, is one of remarkable adaptability rather than fundamental difference. Every population studied, whether Chinese, European, or any other group across the globe, descends from the exact same relatively small population of early humans who began migrating out of Africa within the last 100,000 years. The genetic variations researchers have cataloged between Chinese and European populations represent tens of thousands of years of separate environmental pressures acting on that shared ancestral foundation, producing a wide range of specific, traceable adaptations along the way. Far from revealing some kind of deep, unbridgeable divide between populations, this research actually reinforces just how recently, in evolutionary terms, all modern humans shared common ancestors, and how remarkably similar our genomes remain despite tens of thousands of years spent apart on opposite sides of the planet. Make sure to like this video if you find these deep genetic mysteries as compelling as we do, because there are countless more stories like this one still waiting inside the human genome for scientists to uncover. At the end of the day, comparing Chinese and European DNA doesn't reveal two separate stories of human evolution running in isolation from each other. It reveals one single shared story that's split into two remarkable paths, roughly 45,000 years ago, each shaped by its own unique encounters, environments, and evolutionary pressures, before modern science finally found the tools capable of reading exactly how those two paths diverged and what traces they left behind along the way.